Yeah.
Well, hi, everyone. Good evening. Today I will give you a small, brief explanation over a research article in the topic is synthetic biology. It's about an experiment called Hachimoji DNA and RNA, and a subtitle is a genetic system with 8 building blocks. So what it refers to is by the way, I'm sorry for my awful English. It's like a little bit awful. But okay, there's this group of scientists who, well, they decided that, no, they didn't decide, whatever. So it's a research and experiment based on taking advantage of using of looking at the hydrogen bonds that link um, base pairs in DNA as something that you can, I mean, looking at the hydrogen bond as something that it's not necessary at all, or not so necessary as all of the other things that conform uh, DNA and RNA structures. So based on this premise and this idea of making DNA and RNA with no hydrogen bonds or with a different order on hydrogen bonds, these people made, wait, it's here, um, 2 additional hydrogen bonding patterns that join 2 additional pairs. So what this means is that they designed like extra bases. I mean, like, ATGC. So they did another extra 4. So what this means, for example, is that with 4 base pairs, we only get 12 possible combinations. And this gets it to a whole other level. It's 36 out of orders in which it can be arranged. So, well, these molecules, the new ones, they share characteristics with base pairs, like, I mean, with nucleotides. I mean, they are like mainly the same thing, but just they are, I mean, they have a different structuring what refers to the hydrogen bonding stuff. So the experiment didn't go only to make these molecules and attach them to DNA or incorporate them. It actually goes further. It's about making it possible to adapt to mechanisms that DNA carries on. So, for example, it can carry on transcription, it can carry on translation. I mean, incorporating these new nucleotides to translation means that you can add extra amino acids to any protein that you want. Also, I forgot to explain correctly how does this work. So if you have no hydrogen bond, your molecules have to be surrounded by others who have hydrogen bonds for, like, in order for DNA to maintain its structure. If you have 2 of them, like 2 of these that don't have hydrogen bonds, it's impossible for DNA to go on with its typical mechanisms and conducts, So that's the thing. These additional nucleotides can only participate in any mechanism or any behavior of DNA or any process in the condition that they are surrounded by both sides uh, with, I mean, traditional, let's call them traditional nucleotides. This model of DNA and RNA, it's called Hachimoji DNA and RNA because, well, the term in Japanese is Hachi, which means 8, and Moji, that means letter. So Hachimoji is 8-letter DNA instead of 4-letter DNA. And, well, what else? Mm, well, so far, I don't know if you have any questions or anything. I think you do, probably. No?
Is it a theory or
No,
it's
it's practical.
happening?
I mean, there's even like some, I mean, in the article, there's some experiments with pictures and all of that. Still, I mean, what's important about this is that it's an artificial mechanism that's capable of behaving like normal DNA. Even in some parts of the text, they describe it like alien DNA or something. It's funny, interesting. Any other questions?
Yes. Is there any like practical uses that they mentioned that we could use this 8-letter DNA, like apart from just like experimenting and like replicating? Like, is there
Yeah.
any practical uses that we have planned or something?
Well, imagine that having these nucleotides in action, I mean, like, yeah, adding extra chemicals to DNA structure also means that, as I said before, you can have different amino acids incorporated to any given protein. So that may be the principal use. And also they describe it as some, here it is. Hachimoji DNA was then transcribed to give Hachimoji RNA the form of a functioning fluorescent Hachimoji aptamer. Everyone here knows what an aptamer is? Or, well, an aptamer is a molecule, I believe, that can take participation in transcription or something like that. But what's important about this is what you were saying, Victor. The results expand the scope of molecular structures that might support life, including life throughout the cosmos. So it's mainly for research, and to give us an idea that life can have, according to these experiments, a different structure in other places of the universe with different conditions and stuff.
Yeah.
So it sounds like fantasy or weird, like alien DNA, literally.
Mm-hmm. Yeah, and so in RNA that the thymine is replaced by uracil. Is there any other base
Yeah,
pair of these new ones that are like replaced in RNA?
replaced. Uh, Let me see. It's mm, I believe not, but
So yeah.
well, as it appears here, I think not. Any other questions?
No.
No?
No.
Any
I think
opinions?
it's really cool.
Yeah, I also think that, but the only trouble I have like with this is that it needs like some extra research. I mean, from first hand,
Yeah.
it's difficult to understand. And I don't know, I would like to understand it better. Sorry for the crappy explanation, but it's not so easy. Something else? So we may go on with the other article or something.
Yeah.
Yeah.
Okay. Mm, all of us see that? Yes, ma'am.
Yeah.
Okay.
Yes.
Okay, I'm going to start. Um, my paper is about the inactivation of porcine endogenous retrovirus in pigs using CRISPR-Cas9. And um, this is supposed to move. Well, I don't know. Well, in general, porcine organs are similar to human organs in function and size. These animal organs can serve as skin graft, cornea transplant, or bone transplant. In general, they serve as a pre-gene transplant that will provide organ function to patients with an organ failure. Although all these good characteristics of porcine organs have presented, they have an immunological incompatibility with humans, and that is named porcine endogenous retrovirus transmission. Um, and they are called PERVs. PERVs are a gamma retroviruses found in genome of all pig strains that can be vertically transferred through their inheritance. And although there is no study in which the PERVs has shown any transmission to humans, there has been a clinical setting that the PERVs have a highly possibility to infect humans cells and to integrate into the human genome. The PERV integration could potentially lead to an immunodeficiency and to cancer. Well, the purpose of this paper was to demonstrate the method to inactivate all the 72 copies of the PERVs in the immortalized porcine cell. And this porcine cell line is called PK15, and it eliminates PERV transmission to human cells. And the method they used was called multiplex genome engineering, and it is an easy and efficient strategy for the generation of the gene, cells, and the organisms. It uses CRISPR-Cas9, and in the paper, it explains that this method inactivate the PERV activity in a primary porcine fibroblast cell line. And they use it through the somatic cell in the nuclear transfer. Also, they intend to demonstrate that the immortalized pig epithelial cells line generate a green fluorescent protein that maintain active and propagate the PERVs to the human cells. And they detect this by monitoring the PERV copies numbers in both population and clones of the green protein. And these generate two other subtypes of PERV called PERV-A and PERV-B. And these subtypes are presented in the infected human cells, and they determine whether the PERV integrate into the human genome or they stay in the episomal of the human cells. Also, well, they concluded that the infected cells of humans can transmit births to previously unexposed human cells with a percentage of 20 to 97%. So in conclusion, births can be transmitted from pigs to human cells and also transmitted among human cells in vitro. And although it is still unclear whether births infect humans in vivo, they, the results in transmission in the context of xenotransplantation, that is whether they can insert an organ into an porcine organ to a human. To work toward this, um, they use a combination of CRISPR-Cas9, apoptosis inhibitor, and a growth factor to generate birth inactivated primary porcine cell lines. Do you have any question?
Let me think about it.
Okay.
Well, the first thought that came to my mind was whether or not it was, well, I don't know, healthy to have like a porcine cell organ in a human body, like considering the diseases that these cells are prone to have, or I don't know. It was the first thought that came to my mind. Is it possible, or?
Yes, it is possible, but it has like, it can have viruses, but that's why they use the CRISPR-Cas9 because they need to inactivate some retrovirus. Mm-hmm.
Okay.
So you said that, well, some of the effects of these experiments have been tested on human cells, but in vitro, right?
Yes.
So they been tested in vivo? Like,
No,
right?
they do not have any studies of that.
Okay. And is there a plan like to do them or something?
In the paper, they don't know if it will be like a great idea, but
Like ethically adequate or something?
no, they don't have anything like that. So meanwhile, it's a no.
And well, one more question.
Okay.
What's your position? Like, do you think this is like a good idea, a good proposition for the future, or something that can get handy or something?
Yes, well, I like it. I like it because they have the idea to use organs. If you have a failure with, I don't know, any kind of organ, you can use like a reposition.
Yes, but
Okay.
that would be idealistically, right? But like from your point of view, like realistically speaking, do you think it would be viable to apply this in vivo or not?
Yes, well, I think that yes, because in the paper said that the person, well, the pigs are generated, well, are mated really fast, so that it will not have any consequences with the mate.
I want to say something. So, do you think like we're gonna take like, or for example, like take the lung from a live pig, or that we could use those like pig cells to grow like the separate organs? Like, we could have like a bank of 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 like lungs generate from the pig cells instead of, and just have them as backup, and eventually like use them as animal or for humans, or that we have to take them for like, or we have to like take them from a live pig, or yeah.
Mhm.
Okay. Okay, so well, I think that yes, we can take the organs because the pigs have the similar strands, and also they have a similar um, sequence, well, amino acid sequence that was in the paper. But the problem is that they will infect the human cells.
So, continuing the question that Victor just made, if you had a bank of organs like created from the pig's cells, wouldn't it be in that case better just to have a bank of organs created
With humans.
like from human cells?
Can you repeat the question?
I mean, if you're going to generate tissue and organs like in vitro and all of that, so if you want to make it from pig cells, uh, what would be the case? I mean, I think the real thing here is like literally extracting the organs from pigs. If you do like in vitro, it's like it would be meaningless. I mean, in that case, maybe
Wait, maybe it's more expensive.
that's right. Okay.
Maybe like I don't know what you think, but in my opinion, maybe it's like more expensive, or people feel less like
Yeah, I mean, I would also feel weird if
they're not using like another human,
Mhm.
or yeah, I don't know. I don't know what's your opinion on this.
I think you're right. I mean, you got a point.
But what do you think it's more expensive to do it in vivo or what?
For humans.
Uh-huh.
I think in vitro it's more expensive.
No, it's in vivo, no?
Well, I mean, if you got to
Yeah, in
eliminate
vivo.
Transplant?
it would be more expensive, really?
Yeah, I guess, but yeah, I don't know. I'm just exploring
Okay.
Okay.
the options.
Yeah.
Any other questions?
Like if you're gonna do it in vitro and you're cultivating cells, why not just do it with human cells?
Exact.
Mhm.
Maybe it's harder. No, I don't know. I don't know. I'm just. It'd be
Maybe these
cool to
people are
have.
just excited with their experiment or something.
Yeah.
Mhm.
I mean, in that case, it would actually be better to just reproduce human cells, right?
Yeah. Yeah.
Yeah.
Yeah.
Yeah. I agree.
Anyone has any more questions or
Uh-huh.
something?
No?
No.
Well,
No.
that's it.
Okay.
Okay,
Great.
pause. Bye.
How do I end this?
Yeah.